Morning, everybody. Okay, so um, today I'm going to take you through a brief introduction to what clinical research is and what clinical data management is, how clinical trials are helping each and everyone who is living in this world, whether it is to overcome diseases, whether it is to diagnose diseases, or whether it is to follow a particular treatment regimen. And finally, we're also going to discuss about the career developmental paths that you can opt for while you are part of the clinical research industry, particularly in the clinical data management field. Okay. So first, let me introduce to you myself. So I am Gaurav Saha. I am a lead project manager and an assistant manager at renowned CRO. So I have more than 7+ years of experience in this particular domain, and I started my career with Accenture. So there I became a team lead, and then I moved off to IQVIA, where I became a project manager. And now I have taken up the role of an assistant manager and a lead project manager at a leading CRO as well. I'm currently based out of Bangalore. And yeah, so that's about it. Okay, so starting off, the basic definition of clinical research is that first of all we need to understand what clinical research is. Okay, and then only we will be able to move on to what clinical trials are and how does clinical data management play a role in conducting a clinical trial or conducting or helping in the clinical research per se. Okay, so clinical research is basically it's a branch of healthcare science. Okay, and it determines the safety and efficacy or effectiveness of the medications, devices, diagnostic products which are intended for human use. Okay, so in interviews you will a lot of time you will get a question like what is so unique about unique clinical research. So when you are trying to tell about the uniqueness of clinical research, you can always mention that we are in clinical research domain, we are always working towards achieving two things: safety and efficacy. And safety and efficacy of what? Of any medication or any medicine that we are going to consume, of any medicinal product or devices or diagnostic products that we are going to use. And ultimately, these products' safety and effectiveness will determine how much it will be intended for human usage. Now, this last four words are very important: the intention for human usage. Okay, because anything that is going to be considered for human usage, that is related to medication or diagnosis or devices, for that matter, will have to undergo a clinical trial or a clinical research process. Okay, so thereby coming to the third point, clinical research helps us to find a new or better way to detect, diagnose, treat, and prevent diseases. Okay, so what are we trying to do? Anybody who is a part of the clinical research industry, we're always trying to find out a better medication. We're trying to find out a better device to diagnose or a better product at the end of the day, which is going to help us in detecting early detection of any disease, which will help us to diagnose any disease, which will help us to treat any disease, and finally, which will help us to prevent any disease from occurring. Okay. Now, to complete any clinical research, to conduct any clinical research, we need to perform certain experimentations. Okay. Okay, experimentations. Now, these experimentations are known as clinical trial. Okay, so clinical trial is a kind of your clinical research study. Okay, the clinical trial is an experimentation that is basically designed to answer specific questions which a drug manufacturing company or a device manufacturing company or a diagnosis product manufacturing company will be trying to hypothesize. So they'll have a hypothesis. Suppose they say that okay, we are trying to create or a medication that is going to prevent cancer. Okay, any sort of cancer for that matter. Okay, so that particular product or medication is going to prevent a certain pathway within the system, as a result of which the proto-oncogene to oncogene conversion will be prevented. Okay, so that is their hypothesis. As I was mentioning that So whenever a drug manufacturing company or a device manufacturing company, they hypothesize that okay, suppose they have a hypothesis that okay, this particular product is going to prevent the uh, cancer from occurring, it will prevent a certain pathway, which will again prevent the conversion of a proto-oncogene to an oncogene. Okay, so in order to prove that their molecule or that their product is going to do that, they have to perform certain experimentations. Okay, and those experiments are basically termed as clinical trial. Okay, and there are different phases to a clinical trial. Okay, you have different segments of a clinical trial, and basically there are four phases. We have phase 1, 2, 3, and 4, which are basically trials or experimentations that are being done on humans. Okay, but the major experimentation that gets done at the end of the day is related to phase 1, 2, and 3. Phase 4 is mostly some that is done on after marketing. Okay, after the sponsor has got an approval that they can release the product into the market. Okay, by approval, what do I mean? Any product that is going to be used upon humans has to be approved by certain organizations. Okay, it can be the government, it can be government aided organizations, it can be certain organizations which are associated with sponsors, as that is your drug manufacturing companies, or that are associated with certain hospitals as well. Okay, until or unless they say that yes, your product is safe. Okay, it's safe for consumption. Okay, and it is effective as well. Until or unless they say that yes, it is fine to be used by humans, none of the drug manufacturing companies, none of the device manufacturing companies can actually release the product into the market. Okay, whatever medications that you see in the market currently, okay, are there because the regulatory bodies, so these approving bodies are known as regulatory bodies. They approve the fact that yes, this product is okay and can be used for human usage. Okay, until and unless that comes in, that approval comes in, none of the sponsors, so sponsors is also basically drug manufacturing companies or device manufacturing companies, they cannot release any product into the market. Okay, so that is your clinical trial at the end of the day. So basically, clinical trial is not only trying to release new drugs, but also new devices or new diagnostic products as well. Okay, so as I mentioned, there are a total of 4 phases. Now, all these phases are known as the clinical phase. Now, there is something called a preclinical phase as well. Okay, preclinical phase are basically laboratory studies that are done. Okay, researchers across the globe. Okay, they are working either in vitro or they are working in vivo in animals. Okay, animals in the sense that we can have rodents, we can have non-rodents as well. So, can anybody tell me what all animals are included in rodents?
The mice,
Okay,
guinea
anything
pig,
else?
Guinea
Guinea
pig.
pigs,
Guinea pig.
which is similar
mice,
to
guinea pigs, and
human.
sorry.
Which is the mechanism which is similar to human. We can't directly input the human, so in lab condition to uh, 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 we have to study for
Perform experiment
it
on,
exactly.
yeah, yeah, right. So as you said, mice, guinea pigs, even
Rabbit.
rats,
Rabbit.
rabbits, yes, yeah, so these are all
Monkeys.
rodents. No, monkeys will
Monkeys.
be
I know that.
non-rodents. Okay, they are
Yeah.
actually not rodents. They are animals. They are, so we have animals. Under that, we have rodents and non-rodents. Okay, rodents are, as you mentioned, guinea pig, mammals. Sorry, rabbits and rats, mice. All these are rodents. Okay, and if we talk about non-rodents, then we have monkeys, chimpanzees. Okay, so these are your non-rodent category as well. Okay, and in vitro, if I say any experimentation is being done in vitro, what do you understand by that? Question is, when we say we are doing experiments in vitro, what do you understand by the term?
Lab conditions.
Like
Lab
things like
condition.
studying, yeah, bacterial, animal, human cell culture.
cell cultures, right? So it's basically under laboratory conditions when we are using cell cultures, basically conducting experiments on a petri dish or on a, in fact, on a test tube per se, right? Everybody agrees? Okay.
Yes.
So these are your, preclinical environment when we are experimenting upon animals, when we are experimenting upon cell cultures. These are your preclinical conditions. They set up the base depending upon which human experimentations will be conducted. Without conducting this preclinical step, without conducting this preclinical phase for that matter, none of the drug manufacturing companies can go off to this clinical phase. They cannot experiment or they cannot do experimentations on humans. After doing the preclinical experimentation, they have to go for an approval process. Okay, that approval process, this is just for your knowledge now. So I'll just tell you the terminology. It is known as INDA. Okay, that is Investigational New Drug Application. That is the first approval process that the drug manufacturing company has to undertake. Now, once they get that approval, then only they can start off with the clinical phase. That is phase 1, followed by phase 2, then phase 3. And finally, after phase 3 has been completed, they again have to go for another approval with all their human study results. Okay, so in the INDA, they had to submit the results, they had to submit their observations that was there in the preclinical phase and the plan for the human phase. In the after phase 3, they have to go for another approval that is known as new drug application. It is not INDA, but it is known as NDA. Okay, they have to go for this new drug application. Once they submit this new drug application, the information that they have got in the preclinical phase, that the information that they have got in the phase 1, phase 2, and phase 3, all of them will have to be submitted in this NDA approval. Okay. Once this NDA document is reviewed by these regulatory bodies, by these government bodies, they will say either yes or no, or they will advise for some modifications. If they say yes, then only the drug manufacturing companies can market the product and can release the product into the market. Okay, it will be available for sale. Okay, and once that is available, there is a certain span of time when they have to observe the standard effects of the particular medication on a long-term scale. Okay, you will have suppose whether there is, suppose when the particular medication is being used for more than a year, okay, or around 2 years, then are there any long-term side effects coming up within the human body? That they have to observe. Okay, and that can only be observed once the marketing has happened. If there is no such strong side effects in the particular medication because of the particular medication, okay, then it's fine. Then the post-marketing surveillance gets done with. Okay, and during this particular post-marketing surveillance, each and every um, time that they see a adverse event, that they see a side effect, they'll have to report it to these to the regulatory agencies, to the government as such. Okay, if there are a lot of side effects happening, then the regulatory body and the government will ask the sponsor to take back the medication from the market. They need to take it back, and a circular will be issued that none of the pharmacists, none of the pharmacies, hospitals, doctors are supposed to prescribe that medication. Okay, this is the level of stringency that is applicable in the clinical research industry. You cannot release a product into the market that is showing strong side effects in the human population. Maybe till phase 3, whatever experimentations they would have done, that wouldn't have showed the original side effects. But if side effects are coming up after it has been marketed, they need to take it back from the market. Okay, so this is the level of stringency that exists in the clinical research industry. Okay. Okay. Before we go into further details, okay, we need to know what exactly is a drug. Okay? Now, a drug is basically any substance that causes any change in the organism's psychology or physiology when consumed. Okay, so a drug is basically a substance that will be termed as medication when Medication, when it is causing or when it is treating, curing, or preventing or helping in diagnosis of a disease, or it promotes well-being. Okay, a pharmaceutical drug or a medication, that is the normal medicine's work. Okay, but a drug which causes, but we can also classify narcotic substances, but we can also satisfy those substances which we call as drugs, in reality which we call as drugs, are going to change the physiology and psychology as well. Okay, because both the substances causes changes in the psychology of the person, in the physiology of the person. If you have fever and you take crocin, then you start feeling better, right? Your psychology changes. That, okay, fine, I have I had fever, my fever is reducing, now I am well, I can go about and do my work. That is also there. Okay, but if you are taking cocaine, if you are taking marijuana, or if you are taking brown sugar, that will also change your psychology, that will also change the state of your mind. Yes, that is for the worse, but still that is a change in your psychology. So if anybody asks you what is a drug, then the first definition should go. You're not supposed to say the second one. Okay, because the question is about defining a drug rather than a medication. Okay, or a pharmaceutical drug. A pharmaceutical drug will help you to treat a disease. It will help you to cure a disease. It will help you to prevent a disease. Okay, but if you have to say a narcotic substance, it does not do that. Okay, everybody got this distinction? Everybody understood about this distinction? Everybody understood?
Yeah.
Okay. Another thing is, another thing related to this is, see, you can do substance abuse of both. Okay, you can do substance abuse of cough syrup. You can do a substance abuse of, Vicks Vaporub as well. So substance abuse can be done with both the things. But again, you'll have to know that when a person is asking you what is a drug, then the first definition has to go. If they're asking you what is a medication, then the second definition has to go. Okay, but if they tell you what is a drug, in interviews, if you want to create a good impression or a proper impression, okay, you can say both the things, like the thing that I said now, defining drug, defining a medication, defining a pharmaceutical drug, you can apply both answers. And then you can say, we also include narcotic substances under the definition of a drug, because they also change the psychological state or the physiological state of a person. Okay, then that will be something like a top answer, if you are able to remember the entire thing while you're giving your interview. Okay, so even if you answer the first particular definition, it is absolutely fine, it's not a problem. But if you're able to give the whole picture, then it's like creating a separate persona of yours in front of an interviewer. Okay, moving on. Okay, so why are drugs needed at the end of the day? Obviously, in order to meet unmet medical needs. Okay, if you have a certain disease that does not have any medication, obviously we will require a drug for that. And we already know, right? We're living in, since all of us have crossed 2020, we all of us know new diseases can crop up anytime. Okay, and all different kinds of news are coming up where scientists and researchers are doing experiments in Antarctica, where they're seeing different life forms, different kinds of bacterial life forms, microbial life forms, for that matter, which are present in the Antarctican ice. Okay, so we never know, the way the climate is changing, the way the environment is changing, new diseases can crop up anytime. Okay, so for them, obviously new drugs are required. Okay, another reason is low efficacy. So everything starts off with a certain version. Okay, when a new drug comes in, it might have a lower efficacy rate as compared to the one that we get after maybe 10 12 years or 10 20 years. Okay, so if you talk about any uh, medication that is used to cure headache, that is used to cure pain for that matter, everything started off at a lower level. Okay, it used to take some time to reduce that pain. It used to take some time to reduce that headache. But slowly and steadily, as we have progressed in technology, as we have progressed technologically, as we have progressed medically, scientifically, we have developed medications that have improved efficacy rate. Okay, when people say, okay, if I take this medication, it will reduce my headache within 1 hour or 2 hour as well. Okay, some people have uh, you know have even said that, okay, in hospitals they'll give you morphine for that matter. Okay, so morphine reduces your ability to feel pain, or it's basically something that is there's like an analgesic. Okay, so it will relieve you of all those feelings. Okay, so we have progressed a lot, so that is why everything started off with a lower efficacy rate, and in order to gain more and more efficacy, we are developing new drugs. Okay, side effects. Okay, we want drugs which have lesser side effects. Okay, I don't want to feel nauseatic when I consume a certain medication. Okay, I have a product in the market that is very much effective, but it does have a lot of side effects. I have to sleep for 5 or 6 hours if I am consuming that medication. Okay, I need a new particular drug that will not have me sleep for 4 or 5 hours. That will not make me feel nauseated. That will not make me feel feverish. Okay, so those side effects need to be reduced. Again, as we are moving, towards better or advancements in medical industry, advancements in the scientific industry, we also want medications or devices to be available at a cheaper rate, right? So we have to develop certain derivatives of the original medicine. Okay, we will also learn about that in the upcoming slides, that we also develop certain medications that are known as generic molecules, that are they are also known as generic medications. Okay? They are derivatives of the original medication, but they're not the original medicine that was available in the market. So sometimes when you go to your pharmacy, you will see that they'll say you want a particular medication. They'll say, no, I don't have this. But in the same composition, I have another molecule. Okay, I have another medication available. You want to buy that? That's at a lesser cost. Okay, so that is known as the generic molecule. So the original drug that was released might have costed maybe somewhere around 20 rupees per tablet. But the generic molecule will cost you only 10 rupees per tablet, because that's a derivative. A lesser investment has been done in order to develop that medication. Okay. We are going to learn in the next few slides why it costs less. Okay. And finally, obviously, to sustain industrial activity. Okay, so clinical research industry, one thing that we used to joke around when we had started off in with our work in this particular domain, that none of the pharmaceutical companies, for that matter, it may be related to your okay, it may be related to any disease, it may be related to any diagnostic products, medical, biomedical devices, maybe related to anything for that matter. They don't actually want people to get well, right? So that is what you used to joke about, because if people really get well, they get rid of any particular disease, then the business of pharmaceutical companies, then the business of pharmaceutical industry reduces. In fact, if that happens, then we will go out of job, right? All of us, like, who are striving towards this clinical research industry, we are striving towards going into this clinical research domain. So none of the sponsor companies, none of the drug manufacturing companies really want people to get well. They want people to get well temporarily, obviously, so that they see that, okay, this, if I take this medication, then I'm getting well. That is fine. But they really do not want the world to get rid of diseases. Okay, so yeah, so in order to sustain industrial activity, obviously new drugs are required. Okay. Okay. Let's look into this picture rather than this. I will take you through this particular picture in a separate session, but I think this is a better picture for you guys to understand, because this is your first day today. Okay, so everything related to clinical trials is associated with drug discovery. Okay, until or unless the drug gets discovered, we will not be able to perform any clinical research. Now, this drug discovery process has a lot of steps associated with it. Okay, once drug drug gets discovered, it is not a one molecule that gets discovered. At the start of a preclinical phase, we'll have 10 to 20,000 molecules that have potential capacity to relieve any person of any disease, relieve any person of any symptom. Okay, there will be 10 to 20,000 molecules. Out of that, at the end of phase 3, we will have only one molecule that will show maximum effectivity and least amount of side effect. Okay, what we are trying, we as a sponsor or the drug manufacturing company is trying to achieve is a one particular medication that is going to show optimum efficacy and optimum safety. Okay, so at the end of the day, they should be, suppose I'm just giving an example, they are supposed to cure or relieve a particular person of headache maybe within 1 hour, but when consuming the tablet you will not feel drowsy, you will not feel nauseated, you will not feel not have any skin blisters appear. Okay, so this is how every company, every drug manufacturing company is trying to develop their product basically. So once they have those molecules, they will conduct preclinical phase, then they will conduct the clinical phase. They will have they will develop through this clinical preclinical phase. They will develop the chemistry, the manufacturing process, and the control process as well. Okay, then they will label the product. They will be given a green signal by the regulatory body. Then it will be manufactured. Finally, the sales and marketing will be done for that particular product. And after that, the post-marketing surveillance starts. That is phase 4. Post-marketing surveillance. Phase 4 is also known as post-marketing surveillance. Like this, each and every phase also has an alternative name. We will know about them in the subsequent classes. So we have the drug discovery, okay, where we identify what we need to do. Again, we over here is a drug manufacturing company. The drug manufacturing identifies their target area. They will identify, okay, this disease, I'm going to target this particular disease. It may be cancer, it may be hepatitis, it may be any bacterial disease for that matter, any viral disease, anything. It identify that target, and in order to in order for their product to work on that target, they developed certain compounds that is known as the candidate molecule. Now, this concentration of this candidate molecule is in tens and 20,000s. Okay, once they start doing all the experimentation, they will start, filtering out on the products. Not all of them will show maximum effectivity and maximum safety. Okay, or optimum effectivity and optimum safety. Some will have very less effectivity and very large number of side effects. Some will have large effectivity but also large number of side effects. Okay, so they will start eliminating those products. They want something that shows large amount of efficacy rate and a lesser amount of safety rates. Okay, of them. So once they filter out on those molecules which have optimum levels of efficacy and safety, of them they will again start filtering out that, okay, I have got 100 molecules which are showing optimum amount of efficacy and safety, but which one does it the fastest? Okay, so then they will start again filtering out among the top candidates. So it's like a tournament that is happening. We have most of the teams playing for the trophy being released into the, being that particular product which gets released into the market. So we have all the teams playing, and slowly and steadily they cross the preliminary stage, the quarterfinals, the semi-finals, and once the final is done, they get released into the market. And even when they get released into the market, they're under strict scrutiny by the sponsor companies, by another team that is known as the pharmacovigilance team. I'm sure you would have heard about this particular domain as well. So pharmacovigilance's job is to do that vigilance of the particular product. Okay, they will always do a stringent surveillance of the industry while that product is in the market. Okay, so that they can identify any safety concerns that appear. Okay, moving on. Okay, so as I was mentioning, we also have certain derivatives of the original product that is known as generic molecule. If you have any biological compound like a vaccine or a monoclonal antibodies, okay, if that is the first time that vaccine has been developed, then that's a biologic product. Okay, and anything subsequently that is being developed from that biological product, which is an exact replica of the biological product, that is known as biosimilar. But if a product is a chemically synthesized compound, like any tablet, like crocin, like any medication for that matter, suppose your cancer related medications, if that is the first time the product is being released, then that is known as the prodrug. Okay, then once that particular or it is also known as the reference molecule, okay, or the reference drug. Okay, once we have a derivative that has been developed from that reference molecule, that derivative is known as a generic product or a generic medication. Okay. Okay. So this is something related to the phase one. So as I mentioned, phase one is basically the first in human trials. Before phase one, there were no phases where humans were involved, basically. So in preclinical phase, as I mentioned, experiments are done in vitro or in vivo, like in rodents or non-rodents. Okay. Phase one is something where first time introduction of the medication happens in humans. Okay, we use a smaller amount of population because we want to see, okay, if the dosages that we have determined through preclinical phase actually pose uh, some sort of safety concern, some sort of side effect, or in that particular smaller population. You don't want to generally go off to a bigger population straight away, right? So you want to if you have an impact, you should have impact on a smaller population. It should affect smaller population at the end of the day. If we don't see any side effects in the smaller population, then we will move off to the next phase. That is phase two. So there's a different kinds of phase one that are there. We're going to learn about it in subsequent classes. Okay, in phase two, it is the therapeutic exploratory trial. We are trying to explore the therapeutic effects of the drugs or the molecules which has been developed. Okay, so we are trying to explore indeed if my hypothesis is correct or not. If I am saying that, okay, this particular drug is going to cure cancer, indeed it is doing or not. If it is going to reduce the tumor size, it is doing that or not. If it is returning the liver function test levels back to its normalcy or not. Okay, that is your therapeutic exploratory trial. We are exploring the therapeutic effects of that particular drug. After phase two, we have phase three, where we confirm those therapeutic effects. That is why it is known as therapeutic confirmatory trial. We are confirming the fact that yes, indeed, my molecule reduces, the tumor size. It cures cancer. Okay, cures in the sense that the cancerous effect, or cancerous tumor cells, get controlled. Okay, it is indeed helping my liver cells work normally. It is indeed helping the liver test results to return back to its normalcy. It is helping my renal function tests. Okay, so this is basically your therapeutic confirmatory trial. And after the phase three is over, as I mentioned, we get an NDA approval, that is new drug application approval. Then we reach the phase four. Phase four is basically your post-marketing surveillance trial, or PMS. Also, it is known as PMS. Now, during this particular trial, there is no fixed number of subjects. It is the job of the pharmacovigilance team. The clinical data management team will not be involved in this particular phase because the product is already in the market. The pharmacovigilance team is going to do a strict vigilance of the market and see if there are any side effects cropping up. Okay, the side effects which were not previously mentioned by the sponsor company in their NDA. Okay, now we will see that, as I mentioned, right, that we always filter out. It's like a tournament. Not all the molecules will be released in the market. So there are various reasons which causes or which induces drug failure. Okay, it can be toxicity for the as the most primary reason. Basically, it creates safety concerns. It is harmful for humans to be used at the end of the day. Okay, you are taking a medication, but instead of getting better, you are becoming more and more sick. Okay, whether it has carcinogenic effects on a long-term basis or not, that also has to be checked in animal, obviously in animals first, and then only it will be released in humans. Okay, the efficacy rate, it is taking days on end to get cured from that particular molecule. Again, it will not be used. Okay, PK properties and bioavailability. Okay, PK is basically pharmacokinetics. uh, I will know what your backgrounds are in the later half, but I hope everybody has learned or everybody knows about chemical kinetics, right? Some point of your school life, some point of your college life, you would have learned about chemical kinetics, right? So we have those like different kinetics that are there. Phase one kinetics, phase two kinetics, phase three kinetics, right? So similarly, this drug availability within the body is also determined by the process of pharmacokinetics. It is basically what the body is doing to the drug. Okay, the body is the rate of absorption of that particular molecule within the body, the rate of distribution of that particular molecule within the body, the rate of metabolism, the rate of excretion by the body. Okay, all these are being measured through the PK properties, are being measured through the bioavailability properties. And if the bioavailability is less, then obviously, so it's like if you have taken in a particular medication and it gets absorbed very fast, it gets distributed also fast, it gets metabolized fast, and it gets excreted out fast as well, then it does not have enough time to show the therapeutic effects that it was intended for, right? Suppose you have taken in a particular medication and you just excrete it out from the body in the next 1 hour. It did not get time to show the not the symptoms, show the effects upon your upon your body. Okay, again, when I say efficacy, we mean that it is able to cure the particular disease completely. It is basically alleviating the symptoms completely. I'm not saying the rate at which it is doing that. We are striving towards that medication which will do that very fast. But as of now, the PK properties will help you in understanding the fact that if it is happening very fast, then it does not get enough time to show its therapeutic effects. Okay, we are trying to achieve that particular medication which will be able to show the therapeutic effects in a shorter duration of time. Maximum therapeutic effect, minimum time. We're trying to achieve that particular medication. Okay, so again, related to that PK properties and bioavailability, we are also going to discuss about inadequate drug performance. Okay, inadequate drug performance in the sense that a drug is not effective and it does also has safety concerns. Okay, so these are the major reasons for drug failure or a molecule to fail in that particular experimental phase. Okay, so these things you have to know in the later stages. So it's not necessary now because today is your first class. Okay, now coming to one of the most important aspect of clinical research or clinical trials, clinical data management. Okay, if you just simply read this terminology, clinical data management, it basically means you are managing, you are taking care of the data that has been obtained through clinical processes or clinical trial processes. Okay, it is helping you to collect that data, it is helping you to integrate that data into specific parameters, and finally it is helping you to validate the data, whether the data is accurate or not. When we say suppose we have taken the physical measurements of a particular person, okay, before going into physical measurements, can anybody tell me, if I say that, okay, if I'm a doctor and you have come to me and I tell me your physical measurements, what all parameters will come in and come under physical measurement?
Age,
And
gender, weight.
BMI,
age, gender, I will still not put under physical measurement. That is
BMI
under a
index,
separate.
BMI
Height
Okay.
index,
and body weight.
Weight.
Yes. Yes. Height and body weight. These two are the main physical measurements. Okay. BMI index is also determined through height and body weight. Okay. But yeah, that is a derivative. But if I tell you that you measure and you give it to me, you cannot measure your BMI index without getting a height and body weight, right? So it's a derivative
Yes.
of these two, right? Right?
yeah,
Yes.
Yeah. So height and body weight are the main physical parameters, main physical measurement. Okay. If I as a doctor say, okay, I want to measure your vital signs, then what all things will come?
It may be body parameter.
Your temperature,
pulse,
Mhm.
BP,
Blood
weight and
pressure,
height,
weight and height is again physical measurement. Vital signs is, as you mentioned, blood pressure and
pulse,
Pulse.
oximetry,
Yeah, heart
sorry,
pulse.
oxygen,
heart,
oxygen level.
heart. Yes, yes. So all these parameters will come under your vital signs, pulse rate, heart rate, yes, all those things. Okay, so all these data needs to be collated somewhere. This data is a part of your clinical trial data. So this data, this physical measurement, this vital signs is just a part of the entire data set, but this gets collected during the clinical trial process. Okay, now the collection of this data, the integration of this data for different patients who are coming in. Okay, and finally checking the fact that yes, this data is accurate. Okay, if I go and see, suppose as a clinical data manager, I go and see that okay, this person had come in for this particular trial, this particular experimentation, but this person is showing that his height is 6 feet and his weight is 20,000 kgs. I'm just giving an example, 20,000 kgs. Is that accurate? Is it possible? 20,000 kgs. Obviously, somewhere this data has gone wrong. It shouldn't have been 20,000 kgs, but it can be 20,000 grams. Okay, again,
Yes.
I'll cancel that 20,000, like suppose 60,000. Okay, it is not 60,000 kgs. Nobody can weigh 60,000 kgs, right? There was some problem with the unit. The kg is wrong, or they have not given a decimal point. Okay, after 60, there would have been a decimal point. Okay, so we need to validate the accuracy of the data. Okay, we need to make sure that the data that has been collected, the data that has been integrated is accurate as per the standards that are set for a particular clinical trial process. The standards are defined by protocol. Now, a protocol is, so I guess everybody would have read research papers, right? Where they will see title, we will see the author's names, you will see where it has been published, what is the abstract, what is the background, that is the introduction. Then you have the experimental results. Then you have the conclusion, right? Everybody has seen a research paper, how a research paper looks like.
Yes.
Okay. Similarly, a protocol is also developed for a clinical trial because clinical trial is a large scale experimentation process. It's a large scale research. Okay. A protocol that gets developed has to be approved by the government, the regulatory bodies before it can be utilized for performing in experiments. The protocol will have similarly a title which will define the hypothesis. Then we have the sponsor's name, we have the hospital's name, we are not the hospital's name, but the doctor's name who is conducting the particular research. Okay, we have the synopsis, we have the abstract, we have the basic information that okay, what are the subject population that will be involved, what is the stage of this particular clinical trial, whether it is phase 1, 2, 3. Okay, we also have the schedule of the particular trial. So what is the time duration of that particular trial that will be placed, what are the experiments that have been done that will be placed, what are the parameters that will be tested that will be placed over there. Okay, so all these informations will be placed in that particular protocol. The only thing that will not be placed over there is the result. The result will come subsequently and will be integrated or collected in a database. Okay. Now there are different clinical trial databases in the industry currently. We have RAVE, we have OC, we have Inform. Now, all the data gets collated according to the database that the sponsor has selected. And finally, we as data managers, we would have to go into these databases and make sure that the data is accurate and as per the standards that have been set by the protocol. So everybody has some knowledge about standard unit, that is SI unit, standard international unit, and SI unit, SI values, standard values.
Yes.
Everybody has some sort of knowledge about this.
Yes.
Okay. So similarly, in clinical trials, we also have to report everything in standard formats. Okay. In terms of the SI unit, in terms of the whatever results get reported should be in terms of this SI unit. Okay. Without this SI unit, the regulatory body is not going to accept your data. Whatever the hospital, whatever the doctor reports it as, it can be reported in grams at the hospital, but it has to be converted into kgs. It can be reported in G per DL. If the SI unit is G per L, it has to be converted to G per L and then it has to be reported. Okay. So when that collection is happening, the collection can happen in the lab's own unit, but the integration has to happen in terms of the standard international unit. And then the validation is supposed to happen. Okay, so this is basically clinical data management. Now, there are a lot of activities which clinical data management does. So this is the basic, outline where the clinical data management is involved. So basically, a subject is a person, is a patient who has taken part in a clinical trial. Okay, so the clinical trial goes to the hospital. He or she gets his or her blood parameters, stool parameters, urine parameters. Then, if it's a cancer study, then tumor samples are being considered. And you have CG data, we have immunology data. Okay, all these different kinds of data gets collated. Okay, once it gets collected, it gets integrated into a database. Okay, then we do the review of that data. Okay, by reviewing, we are trying to validate the data. And finally, we go in for a database lock. We, as of now, just know by database lock, we are basically telling the sponsor that everything has been checked from our end and everything is accurate as per our knowledge. There's a disclaimer from the data management end. We start off with the data entry, we end at this database lock process where we say that, okay, all the data has been entered. The site has been asked questions accordingly. That is, the hospital has been asked questions accordingly. The hospital has answered all our queries, and we have approved everything from our end. We have checked everything from our end. Then this entire data set will be analyzed by the biostatistics team for statistical significant results, because that is the ultimate proof which tells you that yes, this data is accurate. This data is proving the fact that the drug is working at the end of the day. If I say that, okay, the normal value is between 5 to 10, that is my normal reference range, and a value has been reported as 50, okay, it is way outside the reference range. My drug is able to reduce the value from 50 to maybe a 15. That is a significant change in the result. That is proving that the drug is actually working. Okay, even though it has not been able to bring it back to the normal reference range, but as of now, still it is fine. It is able to reduce it to by a lot and lot. Okay. So that is the statistical significance or clinical significance that the biostatistics team, by analyzing the data, want to prove. Okay, now there are different trial stakeholders who are present. If any trial is getting conducted, we have the pharmaceutical companies, that is your drug manufacturing companies. We have the CROs or the contract or clinical research organizations, where we have the data managers, we have the research coordinators, the pharmacovigilance people, the medical writers, regulatory affairs managers. Then we have the regulatory bodies, that is in India, the regulatory body is CDISC, and the chairman of that particular organization is known as the DCGI, okay, Director Controller General of India. Okay, so then we have the ethics committees, we have animal ethics committees, we have human ethics committees, we have the hospitals, we have the accredited laboratories. Okay, only accredited laboratories can take part in clinical trials. Everybody knows about accreditation. Has anybody heard about accreditation? Does anybody not know what accreditation is? Can anybody tell me what is an accreditation?
It is like a recognition to carry out some particular test, like a specific type of test.
It's like an approval form, right? It's
Yeah,
a
approval
certificate
form.
to
Yeah.
approve that okay, this particular lab can conduct this
So
experiment,
like
right?
chemical, biological, mechanical, all
Uh,
companies can.
yes, right.
Like
So
also.
if I have to give you an example, if I have to give anybody an example, I always give an example of a college. Okay, a college has any college will get in AAC rating, right? The NAC
Yeah.
rating that they get, they will say A plus accredited or A accreditation, right?
Mm.
Similarly, all hospitals and all laboratories also have their own accreditation. It basically approves that particular hospital or that particular laboratory on in doing its day-to-day activities. For hospitals, it is NABH. For laboratories, it is NABL. Okay, so it's up to you guys to find out the full form. Okay, so for NABH and NABL, that accreditation, that certification is mandatory for a hospital or a laboratory to run its day-to-day activities, and it's basically a certificate that tells that yes, this particular lab or hospital is ranks among the top tier, or it is okay, or it is fair, fairly infrastructured. It has ability to conduct these experimentations properly. All those characteristics get certified by these accreditations. Okay, so these are the different stakeholders who are there in the clinical research industry. Okay, so this is in detail basically.
Um,
Yes.
my question is like there are so many accreditation thing, no organization was there. So which is the particular
Uh-huh.
one used in clinical trials? Is there anything like
No,
that?
no. See, this is depending upon the doctor who is being recruited by the sponsor. Okay, so
Okay.
what happens is it's like an experience, that minimum amount of experience that a doctor needs to have. Okay,
Mm-hmm.
it's like the kind of disease that the sponsor or the drug manufacturing company is trying to address. Suppose they are trying to address any
Mm.
cancer, okay, cancer for that matter. Okay, so
Yeah.
then they will opt for an oncologist, right, who
Okay.
knows
Mm.
about this, who has worked on this particular cancer disease basically. It may be breast cancer, it may be bone cancer, leukemia, anything for that matter. Okay,
Okay.
so blood cancer. So if that doctor has experience in working in that particular kind of cancer, then they will be selected by that particular sponsor, okay, or the drug manufacturing company. Then the
Okay.
lab which is associated with that particular doctor. So any suppose Dr. A is part of Fortis Hospital.
Mm.
Okay. Now there will be a lab that is associated with Fortis Hospital where all the tests will be performed, right? That will be there.
Mm.
Then that laboratory is going to be used.
Okay.
Okay. If that laboratory does not have an infrastructure to conduct any sort of experimentation or any sort of experimentation for that matter, then according to the recommendation of that particular doctor, another lab will be selected. Okay, a hospital gets
Okay.
selected because of that particular doctor, and because of that hospital, a lab will get selected.
Okay.
Okay,
Yeah.
I was able to answer your question.
Uh, so like we can tell like it is like increasing the quality and reducing the error of laboratory, right?
Yes, absolutely, absolutely.
Okay.
So as long as you keep keep to one particular laboratory for one
Okay.
particular doctor, basically, it is absolutely fine, right? So all the patients who are coming into Fortis Hospital will get their tests done at that particular lab which is associated with Fortis,
Okay.
right? So that is how we can reduce the error rate, basically. Okay.
Okay.
Okay. So coming back to the data management
Sir,
activities,
sir, I have one
yes,
question.
yes,
Uh,
yes.
yeah, yes. Sir, for after preclinical study, the four phases
Mm.
are there. Okay. So
Yes,
phase
yes.
1 to phase 3, then we
Yeah.
come to the CDL to validate the accurate data. right?
Yes.
Okay. After that, we have to do post marketing. Okay. So
Yes,
in the
yes.
post marketing study, if the we are doing like in a small population, right?
No,
Not
no, 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 no. Post-marketing study is in a very, very large population. There is no extent of the subject number. We can define the subject number in phase 1, 2, and 3, but we cannot define the subject number in phase 4 or post-marketing surveillance because, it is in the market already. It is being sold. So there can be, hundreds of patients who are taking in the drug. There can be 1,000 patients also. There can be 10,000 patients also who are taking in the drug. So we don't have a limitation of the number of patients who are consuming that drug. Okay.
okay, but this is certain drugs are also banned after post marketing study
Yes,
also,
yes, yes,
right?
yes, yes.
So
Absolutely.
okay, for that also, so who is the concerned person who will direct to report to the DCGI? Who is the
Okay.
concerned person?
So it Okay. So what happens in during the post-marketing phase is, suppose, as you have taken the example of DCGI or the CDISC, basically, who is the regulatory body of India, suppose. Okay. So now in post-marketing surveillance, the pharmacovigilance team is there. The pharmacovigilance team will review for any side effects or adverse effects that are appearing in the patients who are buying that particular medication, and who are getting any side effects because of that medication. They will report that immediately to the sponsor. They will report, the physicians can report that, the patients can report that, but that will get reported to the pharmacovigilance body. The pharmacovigilance body will in turn send it back to the this regulatory team or the sponsor as well. Okay. And sponsor, if it is being sent to the sponsor, then the sponsor has to immediately report to the regulatory body, or the pharmacovigilance team can also report it directly to the regulatory body. Okay, and this entire reporting, the moment it comes to the pharmacovigilance team, it has to be done within 24 hours. Okay. If the patient eats a particular tablet today and he starts feeling nauseatic, suppose after 3 or 4 hours, he immediately has to tell it to his doctor who has prescribed it. Okay, the doctor will immediately report it to that concerned pharmacovigilance team. Okay, and the pharmacovigilance team within 24 hours will have to report it back to either the sponsor or the regulatory body. Okay, so this particular team is actually the major player in reporting side effect in about a particular medication. And once that regulatory body comes to know, they start analyzing the data. Okay, so they will start reviewing, okay, how many side effects have appeared. Suppose in a population of 10,000, only one subject said one time that I am feeling nauseatic. Okay, then it's just a side effect that has appeared, maybe because of some food that you would have consumed that day, maybe because of some lifestyle change that would have happened for that particular person. Okay, but if out of 10,000 people, 100 people are saying that no, we are feeling nauseated, we are feeling dizzy, we are feeling pukish, okay, then that is a concern. Okay, because that's a lot of people who have got impacted. That's a lot of people who have been basically borne the brunt of that particular side effect. Okay, as a result of which the regulatory body will, again, the same process follows. The sponsor or the pharmacovigilance will have to report it to the regulatory body. The regulatory body will basically tell the sponsor that no, this is a lot of side effect, a lot of side effects that are happening. You need to take it back. You need to take it back from the market. They'll basically ban that particular product. They will say that until and unless they review the data, they review the experimentations again, and they release again, send it through that clinical phase, the phase 1, phase 2, and phase 3, without any side effects. They cannot release that particular product.
Okay.
Okay.
Thank you.
Yes. Okay. I was able to answer your question.
Yes, sir. Yes.
Okay. Fine.
Yes, sir.
Okay. Okay, as I was saying, the clinical data managers have their entire task divided into three, departments. One is startup phase, where you are creating the database, or where you are basically design the basis on which you will have the data entered. Okay, or you will you're basically creating the framework of the entire setup, basically. That is a startup phase. Then you have the conduct phase where the data has started entering. Okay, the patients have started coming into the trial. We start reviewing the data, whichever is coming into the trial. We start validating the data. Under that, there are a lot of tasks that need to be performed. Some of them have been listed accordingly. Then we have the closeout phase where I said, right, database lock, that is the closeout phase where we are we have completed validating the entire data set, and we are basically saying that, yes, we are done from our end. Okay, so these are the three phases. The major task that a data manager does is to rectify any data discrepancy. Okay, you will always hear the every organization that is there, if you are a part of the data management team, you will see, okay, I have these many discrepancies, I have to resolve these many discrepancies, I have some doubt in this discrepancy. They will always be talking like this. Now, data discrepancy is something that is a part and parcel of a data management's life. Okay, data manager's life, basically. So any data that is not legible will or not complete is considered to be data discrepancy. We'll just see the types of discrepancies. So if a data is missing, if a data is illegible, if data is out of the normal range, if there is any date related issue, or if there is any logical issue with the data, okay, then that data is termed as a discrepant data, or there is discrepancy in the data, as a result of which it is our job to make sure that the data gets rectified. Okay, the discrepancy is resolved. Or if a discrepancy exists, then why does it exist? It is our job to find that out. Okay, and once we find that reason, we'll have to try our level best to get it resolved. A value, suppose hemoglobin value. What is the normal value of hemoglobin for a male person?
12 to 16.
12 to
14
100.
to 16. 14 to 16.
I'll assume it to be around, yeah. So as everybody said, so suppose around 13 to 16. That is the normal reference range, right? What is the unit? When you say, no, unit. If you are saying 12 to 16 or 14 to 16,
Grams
what is
per
the unit? That is
DL.
the value. Grams per DL, right? G per DL. Now
Yeah.
the value of a particular uh, person is suppose 20. Okay, suppose it is 20. It is outside the normal reference range, right? So my normal reference range is 12 to 16, suppose, and its value is 20. It is outside the reference range. So I, as a data manager, will have to question this result. Like, how did it become 20? Okay, why has it been reported as 20? I'll have to ask the question to the lab who has conducted the experimentation. Okay, they need to take back their documents where they would have documented that result, and they'll have to report it back to me that, yes, this is the result because of this reason. They'll have to tell me the reason behind that. Okay, and only after looking at the result can I decide that, okay, it is correct or not. Okay, I cannot change the data from my end. Okay, I, as a data manager, I do not have the power to change the data from my end until or unless the site corrects the data. Suppose they were supposed to write 12.0 to 16.0 G per DL. Instead of that, they just mentioned 140. They forgot to put the decimal point. I know that they have forgotten to put the decimal point, but I cannot go and put that. I will have to ask the lab to do it. Okay, I know that is an error because of the decimal point. It is not because of the unit, but it is because of the decimal point. I'll have to tell the lab that, see, I found this error. The value is way outside. You please check back your document and update the correct data. Okay, they will check back and they will update the correct data. Okay, so this is a type of data discrepancy. Okay, now there are different stages in the clinical trial. Now this is irrespective of what phase you are in. You have an approved protocol. The protocol will tell you which phase the study is in. Okay, what is the kind of disease it is addressing, what is the kind of experimentation that you're going to do. Then the investigator has to be selected. Investigator is basically your physician, your doctor, who is going to conduct that clinical trial in a certain hospital. That particular investigator has to be recruited. Then the approval process will start. The approval process, as in the INDA approval process, will start. Then enrollment and ICF. ICF is informed consent form. ICF is informed consent form. This is one of the major and most important tasks in clinical research. Without a consent from the patient, you cannot do any experimentations. You, as in a sponsor, cannot do any experimentation without a proper consent from a patient. Okay? Once the patient gets enrolled, after the consent has been given, then only can they conduct any sort of lab related test upon that particular individual. Okay. Once the test is done, then the data will get entered into a electronic document that is known as CRF. We will just come to know what a CRF is in the next slide. Then we will have to review that particular data that is present in the CRF. We will have to make sure that everything is validated. We will have to lock the database, submit it for data analysis. And after the data analysis, there is a review meeting from the sponsor end, and then they will file in for the approval, FDA approval. Okay, after at the end of phase 3, they will submit it for the approval process from the regulatory body, from the government body. Okay, so these are the different stages in a clinical trial. A case report form, as I mentioned, is any printed or an electronic document or a questionnaire where the clinical trial research data will be captured. Okay, that can be associated with your lab parameters, which will again include blood parameters, urine parameters, stool parameters. Okay, under the we have the ECG data that will be documented. We have pregnancy related data which will be documented. We will have pharmacokinetic data, tumor related data, vital signs, physical parameters. All this data will get documented in separate case report forms or electronic documents where this data will be integrated or collated. Okay, and annotated CRF, on the other hand, is just a different, just for your knowledge as of now, we will discuss in details about what annotated CRF is. Okay, so this is basically a particular document that has annotations which defines what exactly needs to be filled in each of the fields that are present. Okay, so this is a CRF, suppose. So this is a CRF, okay? And annotated CRF will have something like, I'll just show you. And something like this, an arrow will be there. Okay, and we will have a box, and we will mention within this box what information needs to be updated in this particular field. Okay, contact number is the field. We'll have the arrow, and we will have it written PDF copy that will be there. And we will have all the information present that, okay, under this each and every field, what all information is required. Okay, or what all corresponding variables that can be used. So we will, so whenever we go off to the creation of any data set from this, so it's a very detailed thing that we do. We don't, when we review the data, we don't review it in the database itself. We extract reports from the database. Okay, we extract reports like when we cut a ticket in any portal, we will extract a report, right? That, okay, I want a printable copy, a PDF will get generated. That's a kind of report. Instead of a PDF, we generate it in an Excel format. So that sort of an Excel is presenting the entire data of the database in a nutshell. I can review the entire data of the database in that Excel sheet itself. Okay, that is known as a data set. So you can have a demographic related data set. So demography related data set includes your gender, your age, your race, ethnicity. All these informations are there. If you are pulling a physical measurement or a vital signs data set, then you will get all those information of each and every person who is part of the clinical trial. We will get the physical measurements and vital signs. If we bring up the ECG data, we'll get the ECG data. If we bring up the tumor data, then we'll get the tumor data. Okay, accordingly. Similarly, we have to review the data for each and every subject, or even each and every patient who is part of the clinical trial. Okay. Like this, we will conduct the review of the data. Okay. So this is basically a view of the case report form. You get the history of the particular subject, the medical history. Okay. Does he have any smoking history, alcohol history, burning, amputation, heart attack, stroke, diabetes, anything for that matter? Any family history, personal details like height, weight, waist circumference, BMI, what is the education? Okay. So these are all visits. These are the visits are any time the particular subject goes to a hospital to get his or her samples analyzed, or drugs need to be administered within that. Anytime that person goes is termed as a visit. First time the person goes, it is visit one. Second time the person goes, visit two. Okay. Date of visit is the date when the person had gone to the hospital. Okay, so what are the blood related parameters? Okay, what are the dipstick parameters? Dipstick is again related to your urine parameters. So these are the different parameters that will be documented in a case report form. Again, the case report form will be created according to the protocol. Okay, so these are the different things. Now the reporting timelines, okay, you don't have to know this as of now, but as I was mentioning, right, in the post-marketing surveillance, everything needs to be reported within 24 hours. Sponsor can report it to the regulatory body within 15 calendar days, but the sponsor needs to be reported within 24 hours, or if you are reporting directly to the regulatory body, then you will have to report it within 24 hours. Okay, the sponsor can take some time, but all other stakeholders, whoever is reporting either to the sponsor or to the regulatory body directly, will have to report it within 24 hours. Okay. Okay. Now coming to the most important aspect, I think everybody would have had some sort of a doubt about this. So what are the career levels in basically your um, clinical data management? Okay, whenever you start off uh, whenever you start off with clinical data management, you enter as an associate level. You enter in an associate level. Okay, that's the entry point. Now after the associate level, you go into an analyst level. Okay, then you slowly move on to senior analyst. You become a specialist, you become a project manager, you become a lead project manager. Okay, then you become a project lead. Okay, it's not a lead project manager, but it's like you go into the sponsor's end basically. Okay, you go and work for the sponsor, that's the project lead. Okay, then slowly and steadily you become a principal, a senior principal. Now these are the different levels basically. So you, if you have to tell it in any organization terms, you can become something like once you become an assistant manager, you can become an associate manager, you can become a manager, then senior manager, then you become associate director, and these are the different levels. Okay, so assistant manager, under that you have a specialist, we have a senior analyst, we have an analyst, we have an associate. So like that, the career levels can progress through in clinical data management. Okay, and in clinical data management, if I have to say, or in the clinical research industry, I will say recession will not hit that much like it will hit an alternate industry. Okay, because I have seen in my personal experience, I have seen, and I know even Pankaj and Sridhar can back me up on this. Okay, that we have not really seen people lose their jobs ever because of any recession that has been hit. Okay, it's one of the most stablest jobs that one can do. Okay, and if you lose a job in this particular industry, that means you have done a grave mistake. Either you have breached your contract, okay, with the organization, or you would have done some sort of a sabotage. Okay, until or unless you do that, you cannot lose your job over here. Obviously your performance also matters. If you are not able to work properly, if you are not able to understand the work properly, it's not that you are a bad employee or something. It's like you might not have been fit for this particular industry, you need to venture out and do something else. Okay, but most of us who are working in this particular industry, at the end of the day, we feel that we actually do something good at the end of the day, because yes, we are helping patients on a real life basis, on a real time basis. A lot of patients are waiting out there to get some new drugs out in the market, and it is our job as data managers, our job in the pharmacovigilance department, medical data reviewers, medical data coders, the regulatory team. It's our job to make sure the correct and the perfect drug comes to the market as fast as possible. Okay, that keeps us motivated, and you will not lose motivation if you start thinking about that. Okay, financials are always there, that is a part and parcel of life, but this is something that will help you. Now, all of us could not become doctors that we are going to do operations or surgeries, right? But this is also another type of healthcare professional, okay, professionalism for that matter. You are part of this healthcare professional industry where you are able to make a difference in the life of a particular person. Okay, now, during this training sessions that I take, I will take you through all different topics that are related to clinical research, clinical trial, clinical data management. I will show you some, excerpts from real-time databases that are there. Okay, I will show you how exactly certain steps or certain activities are getting done. Okay, and you will be able to understand at the end of the day how exactly to give uh, a proper interview in order to get a job. Okay, I'll tell you those important points that you need to remember while you have to give while you go for an interview for that matter. Okay, so yeah, so this is what you can expect in the clinical in the CDM trainings that are there. Okay, and yeah, so these are some of the companies that are currently there in the, in India as of now. Okay, some big shot companies are there. We have some companies like Accenture, TCS, Cognizant as well. We have companies like Paraxel, IQVIA, Covance, APD, Care Health Science, AstraZeneca. All of these are there. Okay, and finally, yeah, so we also have some sponsors, some drug manufacturings also, which are drug manufacturers also there in some of the cities in India. We have Pfizer in Chennai, we have AstraZeneca, we have GSK in Bangalore, we have Eli Lilly in Bangalore, we have Novo Nordisk in Bangalore, Bayer is in Bangalore. Okay, so a lot of sponsors are there. In Mumbai also there are a lot of sponsors. Okay, so there is no dearth of companies as such in India per se. Okay, and time and again on a quarterly basis, opportunities come up and you just have to grab them. Okay, so, and it is my job to help you out as your trainer to how to exactly answer questions in an interview, how exactly to prepare yourself for a particular interview. Okay, I think from here, Sridhar, if you want, you can just take us through the next part of the presentation.
Yeah, thank you, Gaurav. Thank you, Gaurav. Hope you all can hear me.
Yes, Sridhar, you're loud and clear. Yes.
Yeah. All, a very warm welcome on behalf of BCRI, and thanks for your valuable time, and hope you all enjoyed the session. Basically, we have organized the session to give an interactive exposure on what exactly clinical research and data management and how both are linked together. Definitely, it's a career-oriented program where the opportunities are very high, as even Gaurav mentioned, and he has shown few companies also over there. And when coming to the growth rate of healthcare, when compared to any other industry, as Gaurav mentioned, it's a recession-proof industry. I can say definitely it's a recession-proof industry because drug design, development, clinical trial, clinical management, clinical data management, pharmacovigilance, analysis of the data, it's a continuous process. It's not about only COVID. COVID is just for some period. We have oncology treatments, we have diabetology, ophthalmology, many developments have to be needed. That may be for a drug, device, or for anything. So clinical trial has to happen. When clinical trial is happening, lot of employment is getting generated. that definitely we have very good amount of scope and quality in case of these career oriented programs. And also you can see the growth rate when compared to any other industry. Almost I can say it's vertical. Almost it's vertical. And once again, I want to say about us, we BCRI, Bangalore Clinical Research Institute. Maybe a question from your side, why we need to choose you? Definitely our concentration is very high on quality training, very high priority to quality training. And in the next pattern, try to place the maximum. It's not about taking a program with us and how much extent you are going to be successful in the interview. And also we are able to train you in such a scenario like you can walk into any sort of interview. Even our trainers will guide you how to face an interview, what might be your interview questions, and how to go through with clinical research and developments. Okay, definitely we are going through and we are giving a very good platform for a continuous learning process. Next slide, please, Gaurav. Gaurav.
Yeah, are you able to see the slide? I
Yeah,
have
yeah,
changed the slide. Yeah.
yeah. It has taken some.
Okay.
Yeah,
Yeah.
I can say these are a few of our students from BCRI who are already placed in. I can say if you are working in Covance, if you are with IQVIA, if you are with Accenture, and no way that few of them even doesn't have good communication also, but they have high intensity to learn the things, and few even have a huge gap also, like 2 to 4, 5 years. They have cope up because of their knowledge. They're highly concentrated on their. Take example like Anita, or else Yashashwini, or Vinita, whomever it is, Shifa. And this month, in Feb month, we can proudly say almost 10 people have been placed in multiple companies. I can say any sort of company that has any sort of like I can say Accenture, I can say Covance, I can say IQVIA, Novo Nordisk. So there are opportunities. There are very good amount of opportunities are waiting for you. We are there for to provide a quality training for the people. That can be possible with us. Go to the next slide. Okay. So what exactly you need? As already Gaurav mentioned, once again, I am saying clinical research industry is looking for a professional who knows a basic knowledge, need not to be expertised. You just need to know the terminologies, adverse drug reaction, medical history, vital sign. We need to know some terminologies. They are not expecting everybody should be expertised. No, they will make you expertised. Once for the entry level or to get a job, the fifth point is very, very important. You need to have very good communication skills. You know the subject. You need to communicate to them. You need to inform to them that I know this. So the fourth one is very, very highly required. And the remaining one and all will come once if you enter into the industry. And what we are providing to you is what are the terminologies and how the terminologies are applicable in clinical research and which platform will provide a definite growth as per your profile because few are already working, few are already working professionals. So how this program is going to help to them, that and all we are going to do in our training. And completely all our trainers are more than a decade plus years of experience. So finally, I would like to thank one and all for providing your valuable time. Hope we are going to meet in the training. Go to the next slide. Gaurav. This is just a slide why the people are behind India. Because here we have a large patient pool, very large amount of patients you can see in India. So clinical trials happening. Yeah, you can do it in multiple age groups and different demographic groups. You can do it over here. And also, when compared to experts, very highly available. Let it be IT experts or medical experts, paramedical, pharmacy, whomever. They are highly available, expertise available in India. And when coming to any other country, you can compare the advantages and disadvantages with any other country. Advantages are very high in India. And also we are providing completely the suitable conditions for the companies to start up their completely. A basic startup also can easily make a platform over here. That's why people are looking over here. And also, whatever it may be the thing, look at here, comparative cost advantages. Compared to many countries, we are we have a very good advantage in case of cost. So we have expertized persons, we have patient pool and cost advantage. What else they want? That's why people are looking towards India. And take example of Bangalore itself, we have around 35 to 40 CROs or companies like where the people are providing jobs to us. Next slide, please. Go. Okay, once again, thanks to one and all for participating, and you made the session more interactive. Thank you, Gaurav. Any more queries? Please raise. It's open for discussion.
Yes, thank you, Sridhar. Thank you very much. Yes, if anybody has any questions, please feel free to ask them now. And yeah, if I am able to answer them, I'll be glad to answer them. And if I am not able to answer, then obviously Sridhar and Pankaj are there to answer those
Uh,
questions.
and we are announcing that from Monday, that is on from 22nd, we are going to start the introductory sessions of this batch. That's at 8 p.m., 8 o'clock in the late evening. We are
Yes.
going to start a new batch. For a week will be introductory sessions.
Uh, and about the recordings also, you can tell that everybody will get by default class recordings and the notes.
Yeah. And once the session is over, once the live session is over, immediately you are going to get completely the recordings to your classroom. And also the valuable material will be shared, assignments will be shared, and Gaurav completely will provide you how the interview looks. And some people may be already working, how to develop exactly the skills, what exactly we need to do, completely will be provided. And once the sessions are over, based on the requirement of the company, we will try to arrange the interviews too. And many of the students are already placed. We have quoted only few. Already many of the students have been placed, are getting placed too. So feel free to ask any sort of questions, queries. Yeah, Pujita, any question? Pujita.
Hi, sir. Good afternoon.
Good
Actually, I
afternoon.
have experience in medical coding
Mhm.
in UHG. I'm working in UHG Excel Health in
Okay.
Bangalore. So since
Okay. Okay.
5 years,
Okay.
I'm working as a medical coder. So I just
Okay.
want to shift to clinical research or some like I have done my M pharmacy. So I just want
Uh-huh.
to go in my professional way. I
Mhm.
do. I'm getting bored with this coding, so I just want to change my career to clinical research so that it will be a little curious
Okay.
for that.
See,
Is that
Pujita, actually the thing is you're already part of clinical research industry, being a medical coder. But yeah, so if you want to change your domain from clinical data management, from medical coding to clinical data management or pharmacovigilance for that matter, okay, yes, you can actually do that. It's not a problem. Yeah, you may have to start off with the freshers who are entering, because it will be a new thing for you, right? So clinical data management will be a new thing for you. You might have knowledge about, how exactly the protocol looks like, how what are the different steps that you need to know being a part of medical coding, basically. But yes, clinical data management or pharmacovigilance will be new for you, so you might have to work in that way. But yeah, it will not be a problem at all. I know Excel Health Sciences is in Kormangla, right? Or
yes,
in near
yes,
that.
Adugodi, Adugodi.
Adugodi, yes, 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 right.
Yes.
Yeah, so yeah, I know that, but you don't have to worry about that. You can train
So
yourself
my
over here, and
experience will be considered here if I shift domain.
see
That
your
is what my concern.
Yeah, see, experience will be considered in the sense that you are part of this clinical research industry, okay? But you'll have to start off with, with the freshers, in the sense that your designation and all might be given as, suppose a senior
Question.
analyst, but I know again you'll have to start your training process freshly with the clinical data management team, as in the freshers who are coming into the clinical data management. I'm not talking about the salary or anything. I'm talking about the knowledge part of it. Okay, so we'll have to gain the knowledge along with them. So whatever training that I'm giving it to you, it will help you to crack the interview over there. But once you enter the company, you'll have to learn along with the freshers. Okay, so that
Huh.
is the only difference that will make. But yes, experience and all, in the sense that you, since you're a part of the clinical research industry, it will be applicable. Okay, and I'm not talking in whatever, when I say that, okay, you have to start off with the fresher, I'm talking in terms of learning about clinical data management that is being done by that particular company. Okay, so
Okay.
right, Accenture is there just nearby itself. Accenture also
Yes.
has a clinical data management
Yes.
team over there. So yeah, if Quanticate is there, it's near that Star Bazaar over there, right? So
Mhm.
Quanticate or Accenture, they will take up employees, experienced employees. It's not a problem, because you're a part of the clinical research industry. But yeah, when you enter that particular organization, since you do not know data management that is being done at that particular organization, you might have to work as a fresher. You might have to, so now maybe you are doing QC work of other people, right? So that time
Mm.
you will not be doing the QC work when you enter the
Yes.
clinical data management field. You'll again start with the basic review. That is what I'm saying
Yes.
over here.
Oh,
Okay, if that is fine with you, then yes, it will not be a problem for you to crack the interview and go ahead.
oh, okay. Actually, my concern is like this medical coding work might not go for longer time, longer duration. That is
Because
what I.
automation is happening, right, in the
Yeah,
medical coding process.
exactly.
Yeah.
Yeah,
Yes, right.
it might happen after some years. So this
Mm-hmm.
job may not be for good for longer. So
Yeah,
I
yeah, I
thought
understand that
I
completely.
switch to this domain if it might help me in future. That's what I
Uh-huh.
mean.
Absolutely, absolutely. I understand that. Yes.
Yeah,
And
that
Because,
is
also,
how.
companies.
yeah, and also, Gaurav, when compared to any other, the growth rate is very less. It's not that
Ah.
much when compared to CDM,
Mm-hmm.
pharmacovigilance. The medical coding growth rate is no, not that good.
Yes, yes,
Mm-hmm.
because
You can,
it just saturates at one stage.
Yes,
Afterwards,
yes.
no growth. That is
and also if automation comes, then definitely they'll be.
many of the jobs might go. That is
Yeah, yeah.
Mm-hmm. I understand. I understand. Yes, yes. Yeah, it's not a problem for you. That it will not be a problem. Let's finish this training, and then I'll definitely, throughout this training, I'll definitely tell you those, small intricacies about what you need to say during an interview. Okay, so I'll tell you from now itself, the first question that they will ask you, apart from introducing yourself, is like, why do you want to change your domain? Okay, so
exactly.
then
I
you'll
know.
have, yeah, so that will be one good formal question that they're going to ask you.
Yes, yes,
So,
yes.
and it's not only related to you. If anybody over here is working in a separate domain, it may be in a lab domain, it may be a lecturer, there may be anything for that matter. they will definitely ask you why do you want to change your domain. Whoever is working, basically. Okay.
Mm. Okay, so thank
Sir,
you.
Okay.
hello, sir.
Yeah,
Nilanjana, right?
yeah,
Yes.
Nilanjana. Yeah.
Yes,
Yes.
sir. Good afternoon to everyone. Thank you,
Mhm.
sir. Uh, uh,
Mhm.
uh, my concern is actually currently I'm working at
Okay.
Hester Biosciences Limited,
Okay.
the executive technical services. Uh, my
Okay.
my domain is serology and
Okay.
microbiology also. So my
Okay.
highest
Okay.
qualification is MSc in microbiology.
Okay.
So,
Okay.
so my concern is that like I want to change domain from wet lab
Yeah,
to
that is
this
fine.
technical
Dry lab,
research.
right? Yeah.
Okay,
Yes. Yes. I
yes,
understand that.
and my total job experience is 3 years, 8 months. In
Uh-huh.
microbiology domain, I'm working like since 2 years. Okay,
Okay.
so is it considered or not, or what would be good for me? I just
Okay, so the thing is, I will tell you frankly, your the number of years of experience might not be considered. You might start off as an associate level, but yes, salary wise, they can consider it because obviously they'll ask you, right? So it's like if you are, I'm just giving an example. Suppose you're getting somewhere around 25K as of now, okay, somewhere. Okay, so they will tell you that okay, we are going to give you a hike on that amount, but your designation will be an associate level. Suppose now you are at a suppose as you mentioned, right, the technical expert, right, something is there, but then associate level is something like an your fresher role. Like when you had entered this particular lab, the job role that you had, the designation that you had is the same thing as a counterpart in this particular industry because you will be completely new to this domain. Okay, so you don't have the knowledge about data management, clinical research as such. But yes, you on the financial aspect, they will definitely consider, but maybe not on the designation aspect. You will not be treated as the same person who has 3 years of knowledge or 3 years more than 3 years of knowledge in the data management industry. Okay, it's the same as I was mentioning to Pujita. She might get the designation because she is a part of the clinical research industry.
yes, yes,
Okay,
yes.
being a medical coder, but in your case, since you are not really part of this data management or pharmacovigilance or medical coding industry for that matter, so that is why your designation might not be the same as equivalent to a 3.5 year old. But yeah, your financials will definitely be taken care of by the company.
Okay, okay. Thank you,
Okay,
sir. Thank
yeah,
you, sir.
not a problem.
Hello, sir.
Hello.
One more doubt. Once, uh, as I am experienced, like
Uh,
I'm also considered as a fresher in the case, right? So they won't consider me, I guess. Like I'm
you
a
will not be if you are 5 years at a 5 years of experience. We have senior analysts. Okay, over here, but you might not be given a senior analyst role. You will not be considered as a subject matter expert in the data management part. Okay, you might be a
okay,
subject matter expert in medical coding. Okay, but you do not know about this data
clinical.
management industry, right? Data management
Yes.
part. So you will not be considered as a subject matter expert over here. So you might start off at an analyst level. Okay, you might, but you will be working with the associates, but they will be giving you a sort of a promotion in the analyst level in that way. Okay, in the clinical data management part. But yeah, you might feel that okay, I am going at a lower designation as compared to the medical coding part. But you have to think on the long term basis that yes, once you start learning about this data management thing, okay, if you gain around, suppose the first company is your barrier basically. Once you enter the first company, so this is something that I will definitely tell you in details again. So once you enter the first company, you start working there, you get around 1 or 2 years of experience, then you can
Mhm.
always say that okay, I know and I have a knowledge of around 6 years of experience in this data management part. Okay. Then the second
Okay.
company that you go into, they will be considering that okay, this person has clinical data management knowledge of 6 years of experience, and then your designation will again be at par. Okay, so then you will be moving up faster.
Okay.
Okay, so the first company, you just have to bear with the designation thing. Okay, again, financials is not a matter over there. The designation thing, you just have to take care during the first company. Once you cross that, first promotion that you get, after that, if you move up to another company, then again everything will be at par.
Okay.
Okay,
Fine.
so it's just that the thing is that you just need to change the domain first. So while you're changing the domain, just don't think about your designation at the very beginning. Okay, just think that, okay, I need to change my job role so that I become, at least at the end of the day, my job becomes more stable. And once I have that stable job, learn the process. I will learn the activities that are there in the company, and then I will try to move out and change. A lot of people change over here. Okay, but as you saw, right, I have already changed 3 companies. It's everybody changes companies, and it's not a problem at all. So just try and concentrate on learning the process that is there in that company, the first year or 1.5 year. Okay, the faster you will be able to learn, the faster you'll get that first promotion. Once you get that, then you'll be fine.
Okay.
Okay.
Thank you.
Any other questions, anybody? I know today we could not complete the introductions for everybody. Yeah, I could not complete the introductions today. So definitely on Monday we will do introductions on a detailed manner. Okay, so that is there. But if anybody has any other questions, please feel free to go ahead.
Okay, hope done, Gaurav.
Okay. Yeah.
Okay, thank you, Gaurav. Thanks to one and all for participating.
Thank
See
you,
you.
everybody. Have a
See you
nice
soon.
day ahead. Yeah. Bye,
Okay.
everybody.
Yes.